start with a classical perspective, Hippocrates and Galen. And Hippocrates is often quoted as saying, "Let food be thy medicine." That is in fact a misquote. More relevant is Galen. It's better to understand and treat the patient with food than medicine, because the art of healing comes from nature and not from the physician. With respect to aging, what is really pertinent actually comes from Hippocrates again, and that is there are two things, science and opinion. The former begets knowledge, the latter ignorance. And unfortunately, in the field of aging, we are beset by opinion. So today, there are more people alive aged 60 and over than at any time in human history. So for once, mankind is doing something correct. That demographic is growing. So between 2015, the number of the over 60s goes from 12% of the global population to 22%, about 2.1 billion people. These will have quite a few years of life left, but most of that life will be marred by the presence of disease, because despite being able to increase our lifespan, we have not matched that with an increase in health. So we tend to spend the last 10 to 15 years of our life with one or more diseases. Okay, so global aging. Most of that growing population demographic will be in the global south. As countries' populations increase and living conditions get better. The economic impact of all this is staggering. So in Europe, it's about 1,000 billion per year, or 9.5% of GDP, just for healthcare systems to deal with with age-related disease. And the McKinsey Institute in the US has estimated that if you could increase healthy life expectancy, so that's health span rather than lifespan, you could generate about $6.2 trillion in GDP uplift, frightening amounts of money. And indeed, an increase in life expectancy of just a year in the US is worth $38 trillion, and by 10 years, around $367 trillion. You know, staggering. These figures are all amplified by social deprivation and social inequalities in health. So what we have emerging is a scenario akin to Mandeville's travels. So Mandeville was a medieval British knight from Yorkshire who claimed he had travelled to the Far East and come back with fanciful tales of meeting men with heads of dogs or with heads in their faces, or faces in their chest, Cyclopean Amazonian women, scoliopods who had only 1 foot and they were so big they could shade themselves under it in the desert sun, a medieval bestseller, and a lot of research on aging and anti-aging therapies that are touted in the popular press, often based on very good science, overpromise and are akin to this. So we need to understand how we age at a fundamental biological level to try and improve this situation and to try and educate people appropriately. What we are facing and what we will see emerge with that growing demographic of the aged is the diseasome of aging. So these are all different diseases. They're all treated as separate clinical disease modalities. But what they all share in common is an underpinning component of a dysregulated aging process that precedes their development. The common features include diminished stress defenses, usually mediated by NRF2. That's a master regulator of your cytoprotective responses, a loss of diversity in gut microbes, an inflammatory burden, and a loss of physiological resilience, or frailty. These are features that are all affected by the changing climate we live in as well. Now, for me, an Irishman working in Scotland, this is really relevant because if you look at deaths per 100,000, This is Scotland in Europe, just below Albania. Not doing very well. Scotland is the sick man of Europe, and Glasgow, the city I work in, it's the sick man of Scotland. Amazingly, we have both the best and the worst life expectancy for a male at birth in the developed world. Everyone only sees the bad side of the city, but in the small 12-kilometre geographical radius, we have the best and the worst. So there's conditions there, if I go back, affecting us. Broken down by council area in the city, what you will see here, the picture is skewed by the east end of Glasgow. And this is where social deprivation is very prevalent. And what I mean by social deprivation is not poverty. Poverty's part of it, but it's a lack of opportunity and a lack of facilities. So life expectancy in the UK is currently falling. A lot of the ill health we see is tied to a very steep socioeconomic gradient, and we have to understand why people are aging faster and getting diseases of aging earlier in order to intervene appropriately. So if you want to understand how you age, I'm always reminded of Tolstoy, the start of Anna Karenina. We age like unhappy families. All happy families are alike. Each unhappy family is unhappy in its own way. How we age is highly individual. Aging is also a process. It is not simply a collection of diseases at the end of your life. Aging occurs as an accumulation of deficits taking place in different individuals in different ways with a variety of rates for different organ systems. That is quite a mouthful, but it's actually quite accurate. So aging starts pre-conception and it ends at death. When I say preconception, when you, or a bit of you, is part of an egg in your mum as she is in your grandmother's womb, then your grandmother's environment and lifestyle and her exposures impact your developing mother and impact you. So events can be transmitted epigenetically across generations. So transgenerational, where the effect was not, subject to exposure in that generation, and intergenerational when it was. The whole process is not passive. It's actively regulated by genetic pathways. And if we can understand the basis of those, then we can intervene appropriately and we can find out what's essential for good health in old age. So this is the Shiels trajectory of aging, age 10, 40, and 60. I'm the oldest living male in my family. Most of the men die very young through bad lifestyles. The women tend to live extremely long lives, and I've always berated them for any bad health that I've had as being due to their lifestyle. And then suddenly we've found out that not only are the characteristics for health passed through that maternal line, but they also come via your dad and your granddad. And this is also applicable to humans. And here, we have a number of studies that have shown that high-fat foods and human fathers with phentomycin, human fathers with a high body mass index, have male offspring with metabolic disorders. The mechanistic basis of that is now understood. It's transmitted via sperm-derived mitochondrial microRNAs. So it's coming down both lines. Mechanistically, aging can be hallmarked into features that are common to flies, to yeast, to worms, to mice, and to humans. And these are cellular features and intracellular features such as telomere erosion, mitochondrial dysfunction, epigenetic dysregulation, and accumulation of senescent cells, disrupted autophagy, disrupted proteostasis, etc. And these have all formed targets for interventions using drugs. Amazingly, it's been predicted that if you could cure a major disease like cancer or heart disease, you'd only add 2 to 3 years of life expectancy to a hypothetical 50-year-old woman, because by the time you've developed the disease, you've accumulated significant wear and tear. If, however, you could intervene in the aging process and stop the wear and tear accumulating, then you could add up to 25 years of healthy life. This scenario is not the whole picture. And I'm going to use a crude analogy here. So think of your body like a car. Where we are at the moment is here. When bits go wrong, we can fix it and keep the car running. What we can't do is fix this. When the car is in the wrong environment, so being in the wrong place at the wrong time, exposed to something that doesn't like it, gets trashed. So this is the effect of your environment or exposome. And the exposome comprises the totality of human environmental exposure. So that's both abiotic and biotic, preconception to death. It's a concept that was first developed for cancer by Christopher Wilde in 2004. We have developed that to address aging, and more recently we've now addressed that to the development of future cities. This really looks at how all aspects of your external environment through your life affects your genome and your epigenome, and therefore your trajectory of age-related health. Something that's not easy to model in a laboratory. Amazingly, some of the most recent evidence is shown in the great debate of genes versus environment. Environmental influences make up around 80% of your age-related health trajectory. I still find that difficult to take in, but that's the current evidence. An additional feature of this is the bioexposome, so biopsychosocial effects. How you were raised as a child. Were your parents caring and nurturing, or were they abusive? Did you have overcompetitive siblings? Did you live in a nice, clean, warm house with your own bedroom? Or did you live in damp, overcrowded conditions, exposed to bacterial endotoxins and spores? Did you have good schooling? Access to recreation? Were you well fed? Did you live in a community that was spiritual? Because cortisol-mediated stress responses have a big impact on how you age. And those that practice spirituality of any form tend to live longer, healthier lives. But this whole picture is important because 3 simple exposome factors, namely air pollution, tobacco smoke, and diet, account for 50% of global mortality annually. What I'm trying to do is work within the geroscience hypothesis, and that's designed to get people with a bad trajectory of health and aging improved, to make them more physiologically resilient and physically capable. And to do that, we have developed the kidney as a model of aging alongside Peter Stenvinkle and others, where chronic kidney disease is a disease of accelerated aging, and kidney transplantation is the use of normative healthy tissue therapeutically. And we're trying to work out what environmental factors and lifestyle factors and biopsychosocial factors determine how well you age. And then if we want to enhance someone's physical or physiological capability, we need to know when and where in the life course to interact. Bearing in mind that what's good for you when you're old is not necessarily good for you when you're young. And that is the concept of antagonistic pleiotropy in simple terms. This geroscience hypothesis that's mentioned here is really targeting the upstream effectors of aging and trying to bolt the stable door before the horse has gone. And therefore you want to preempt the burden of lifestyle disease and prevent early access to geriatricians. So it's compressing the period of morbidity into weeks or months and not years. So in Glasgow, what we have observed in our general population is that the key things that matter are socioeconomic position, diet, the epigenome, phosphatemia, again, dad-derived, poor renal function, and microbial dysbiosis. And what we have observed here is that those at lower socioeconomic position that have an imbalanced diet, lacking fruit and vegetables, age faster. They have microbial dysbiosis, they're hyperphosphatemic, and they are epigenetically dysregulated, another sign of accelerated aging. Interestingly, in the context of inflammation, senescent cells, or old cells, secrete pro-inflammatory factors. And in tight laboratory studies, and tightly controlled clinical studies, there's a very good correlation between the degree of senescence and inflammation. In the general population, this gets a kicking. Less than 10% of the inter-individual variation in inflammatory burden is due to senescent cells. And more recently, we've shown most of it's actually due to microbial metabolites. Diet is the most critical modulator of health with respect to age in the general population. That's what we have found. And additionally, that dietary imbalance in Glasgow, at least, is also affected by those that eat red meat too frequently. They become hyperphosphatemic. They have poorer renal function. And that is essentially addressable. To address all this properly and to understand how we age properly, we need to be able to measure how we age appropriately. And that's not easy. So currently, we lack robust markers to measure the efficacy of any interventions. We need to identify markers that can distinguish between normative aging and disease. And most of the markers we have are really designed to track disease. So we need both an odometer to tell us how far on our journey we've traveled, and we need a speedometer to tell us what rate we're traveling at, because we need to know if our interventions can slow down aging. Aging is also not linear, it's segmental, and you age in bursts throughout your life, principally when you're over 40 and over 60. Currently, I'm in Sniper's Alley between 60 and 65. If I get through this, I should be okay. If I don't, then I'll have to address it. There are lots of ways to measure aging, and some of the best are biophysical markers, and these are standard assessment tools such as gait speed or chair rise. One we have been developing, I have developed previously, and applying again, is lens density in the eye. So if this is telomere length in P16, CDKN2A, you can see the correlation with age where lens density is much stronger. It's also a non-invasive measure you can make simply using an iPhone. Biochemical measures such as telomere length, CDKN2A status, skin autofluorescence, phenotypic age, proteomics, metabolomics, microbiome composition are good to a degree. Telomere length is actually very weak. SAF is poor, and I'll show you why. Most of them give you unrealistic estimates of biological age. So this is someone with kidney disease or a transplant. Here at age 20, SAF age shows that they're over 100. Someone at 60, SAF age is showing that they're over 150. Biologically implausible. That's someone with a kidney transplant that we know is of a given chronological age and with good physiological function. Phenotypic age, again, unrealistic estimates of biological age. So the way around this that we have taken is to try and get involved in making epigenetic clocks. And these are based on changes in DNA methylation occurring at given loci as you get older. They are seriously clever tools. Most of them developed by Steve Horvath and his alumni. First developed using chronological age to train the clocks. Then second generation clocks that included chronological age and lifestyle factors or biochemical factors. And then a new third generation set has emerged that they're more nuanced, they're based in big data typically, but not always. And they look at simple wear and tear, so methyl allostatic load. It's a PCR-based 32 loci methylation clock. Dunedin pace to measure the speed of aging. The IC clock coming out of the Buck Institute, based on the general population in Toulouse in France, that's been evaluated longitudinally. And two clocks coming out of my lab that I'll tell you about that are unpublished yet, expose age and the microbiome clock. So for all the clocks, you need to remember that a causal linkage between DNA methylation and aging has not been fully established, and there's good evidence now to suggest that they're not causally linked. There's a limited understanding of how individual methylation ties contribute to the aging process. We have to think about epigenetic drift with age and by exposome factors, the fact that aging is segmental, so there's not a one-size-fits-all, and we need large-scale longitudinal studies and aging intervention trials to clarify the relationships. We started looking at this using individuals with chronic kidney disease and kidney transplant recipients, and could show that if you compare clocks head to head, they're all good as odometers, so they correlate well with chronological age. But what about as a speedometer? We know that if you have dialysis, you tread water, you don't get worse with respect to aging. And if you have a transplant, you do get physiologically better, so you should slow down the rate of aging, and we've demonstrated that previously with telomere length. With the epigenetic clock, what we found was that speedometers, most of them don't work. So the first generation clocks don't really show a change when you dialyze, and they don't show a change when you have a transplant. And even GrimAge, the second generation clock, doesn't do this. PhenoAge from Morgan Levine does, but it is pretty imprecise. And we have made a composite clock called the Glasgow Karolinska clock with Peter Stenwinkel's group that does report clinically and accurately. That clock shows you that the speed of aging reduces once you've had a transplant. And it's predictive of function. So we were very pleased with this. And we've started asking, how does the clock tick? And this brings us back to food. So that Glasgow population with accelerated aging, telomere attrition, hyperphosphatemia, mitochondrial dysfunction, and epigenetic dysregulation, with the imbalanced diet, is more inflammatory markers, principally more TMAO derived from TMA that bacteria in our gut produce, not our cells. But in the same biochemical pathway, the bugs can also produce beta-ine. Now, we make beta-ine in our mitochondria, but our mitochondria become dysfunctional as we get older. So we can replace it either through the activity of bugs or directly via our diet. And what we found out in Glasgow was that our deprived at age fastest, they're epigenetically dysregulated, have less beta-ine in their circulation. That correlates with microbial dysbiosis. And what we observe if we look at bacterial barcodes in the blood is that the deprived that show the fastest aging have more unfriendly gut bacteria or bugs derived from the mouth, they're oral pathobionts, leaking in through the meninges. They have less salutogenic or health-promoting bacteria and less that can make beta-ine. So the epigenetic landscape, unsurprisingly, is affected. So we've now tried to test that clock and make new clocks in the general population, looking at longitudinal cohorts at baseline at +2 and 5 years, and in a validation cohort in Glasgow, which is a cross-sectional study. We've compared them all head-to-head, correlating exposome features, the biochemistry, the biophysical measures, the psychosocial features, and we've tried to create some superior clocks, and this is the unpublished stuff. So I put my head in the block with it. We're looking at odometer measures and speedometer measures for all the clocks that have them, compare them against all the meta variables, and we only take something as significant if it shows significance in at least 3 independent clocks. And then we use the data adjusted for deprivation and sex, and we try to make better composites. The take-home message here is that the clocks, as odometers, correlate well with each other, but they don't correlate well with the speedometers. Interestingly, most of them don't pick up differences in socioeconomic position, but when they do, they're all within a reasonable distance of each other. Hannum reports 2.1 years, PhenoAge 3.2, Glasgow Karolinska 1.9 years. It's a frighteningly small deviation between your chronological and biological age that predisposes to ill health. When you have overt disease like chronic kidney disease, that delta goes to 3 to 5 years. So the other thing we found reassuring was the speed of aging. With deprivation, if you're deprived, it's about 10% or 11% faster, depending on the clocks. There's still a lot of noise. And this is the other thing that disturbs me. And this may be the legacy of trans and intergenerational effects, plus just not having the right tags. But you can see an MAE, or a median absolute error, of 6 years for Horvath. It's large. And we get Our best one at this stage was 3.6 years. We have now improved this with the expose age clock, and it simply combines the Glasgow Karolinska clock with a microbiome clock that we've created. And that has an MAE of 3.3 years, so it's functional. And for longitudinal studies, it's reasonable. But what I'm trying to point out here is there's still a lot of noise, and there's a lot of overhype on the use of clocks. The only one that we have found accurate for normative aging has been the GK clock, and expose age is now being tested. And I'll show you a teaser of where, because we're still in the midst of analysing this, but I thought this was an appropriate audience. As an odometer function, it works well. The odometer also picks up some liver dysfunction, alanine aminotransferase. As a speedometer, the clocks again look pretty good, but we also show that telomere length at a given point reflects the speed you're aging at. And then the speedometer functions also pick out things from diet, how many takeaway meals you've eaten in the past week, how much fruit and veg you're eating, and obesity. My primary read of this, and I'm not the expert in nutrition, is that we may be seeing, as we have seen previously in Glasgow, the effect of inorganic phosphates coming in as preservatives in the food, and a lack of dietary intake of polyphenols because people are not eating sufficient quantities of fruit and veg. And that matches what we have observed in our Glasgow cohort previously. To tidy up now, I'm running out of time. Can we manipulate the exposure of aging? Absolutely, we can. Using food to do so seems really sensible. This paper you've seen from some of my colleagues, particularly Fred Ho and Jill Pell and John Mathers in Newcastle, has predicted you can enhance life expectancy in the UK by 10 years by a simple shift in diet. What I would like to do, therefore, is improve oral health, especially among our deprived, and come up with a new Glaswegian diet, one that's balanced, with less inorganic phosphate, more polyphenols, that can activate NRF2 and support a salutogenic microbiome. So I'll finish with Hippocrates again. Here we are, 3,000 years on, back to basics. Give everyone the right amount of nourishment and exercise, not too little, not too much. We have found the safest way to health. And I'll just thank my collaborators, in particular the group of Peter Stenvigel at the Karolinska Institute, Colin Selman in Glasgow, Denise Mafra in Fluminense, and thank you.